All right, so our essential question, how do cells grow and reproduce? Same essential question as last week. But cells divide during mitosis and cytokinesis. Like I said, it's that M wedge in the cell cycle. Last week we went over the cell cycle. G1, S, G2, and M. And you guys are doing a whole interactive assignment on it this week. And this section is focusing on M. So first we have to talk about what chromosomes are. So a chromosome is one long continuous strand of DNA that is wrapped very tightly around proteins. Proteins are called histones. We don't need to know that yet, but that's what they're called. And they just wrap around and keep wrapping until they end up looking like these X-shaped structures. And there are different chromosomes. Some chromosomes are bigger, some chromosomes are smaller. On the X, those are called arms. Sometimes the top arms are shorter than the long arms. But in general, we draw a chromosome to look like an X. Good morning, Benjamin. The structure of a chromosome. So DNA plus proteins is called chromatin. Remember, we don't say the CH sound. Mitochondria, chloroplast, chromosome, chromatin. Again, I don't know why. We just don't. Half of a chromosome is called a chromatid. And they go by left-right halves. They don't go by top-bottom. Sister chromatids are held together at the centromere. The centromere is that middle portion of the chromosome, the middle of the X. So yes, this is something I want you to draw and label in your notes. That's why at the bottom it says draw and label the chromosome. We also use the female a lot, like sister chromatids. We don't say brother chromatids. Or we'll see in the end, we'll end up with two daughter cells, not two son cells. Again, I don't know why. There's a reason, I just don't know it. Chromosomes condense or get thicker at the start of mitosis. DNA wraps tighter and tighter around those proteins, those histones, and that's what condenses it. So during interphase, DNA is just kind of laying there all willy-nilly. But when mitosis happens, we need DNA to be in those X-shaped structures. And they just keep wrapping tighter and tighter around their proteins, around those histones, until they form the X's. Interphase, the G1, S, and G2 phases of the cell cycle are grouped together and called interphase. During this phase, the cell prepares to divide by growing and replicating its DNA. We went over that mostly last week. An interphase, even though it has that word phase, it is not technically part of mitosis. And if we were in class looking under a microscope at these, it would look like it does in the bottom picture. So that's the picture you would be drawing. Just kind of a dark, all shaded in dot in the center of a square, which is the cell. Mitosis and cytokinesis divide the cell's nucleus and cytoplasm in 4 phases. And in the end, we end up with genetically identical daughter cells. Like I said, we use the daughter. So these are basically clones. And mitosis is the process that 99% of our body cells go through. If we scrape our arm, our body needs to make more cells to replace the ones we lost. Mitosis is the process that makes this happen. Makes the exact same blood and muscle and skin cells that we lost to replace them. Our body is constantly replacing our cells. Prophase is the first phase of mitosis. And chromosomes in the nucleus, they condense. They wrap around those histones and they get thicker and they form those X-shaped structures. Centrioles and spindle fibers form a kind of net. Centrioles we learned way back in chapter 3 when we talked about all of the organelles of a cell. And the nuclear membrane, the membrane around all the DNA that holds it in the nucleus, it goes away, it disappears. So on the left is a GIF, is an animation of what happens during prophase. So let's wait for it to start over. So like I said, DNA is all willy-nilly. It condenses, it gets thicker, and then it makes those X-shaped structures in the middle. And on each end, these are the centrioles. Now, under a microscope, this is what it looks like. So to differentiate it from interphase, this one up here will be interphase because all that DNA is just laying in there all willy-nilly. You can't really tell. During prophase, they get thicker and thicker, and no, they don't technically look like X's, but that's what it looks like under a microscope. Metaphase is the next phase in mitosis, and chromosomes line up individually in the middle of the cell. You can see that happening on the left in the animation. That is called the metaphase plate. And the individual part is important because our cells go through another process where they pair up. They don't line up individually. But we'll get to that next semester in chapter 6. The net, which is all these, those are the centrioles and the spindle fibers. It spreads out and it catches each chromosome right at the centromere, right in the middle. That one on that side, that one catches it on that side. And there's a purpose behind it. And again, under the microscope, kind of just looked all smashed together, but you can tell they are lined up in a line. And it's the microscope pictures that will be on your final next week. So that's why I'm giving you those pictures later. Phase 3 is called anaphase. And like we said, they attached at the centromere, so now they get to be ripped apart. So sister chromatids, those left and right halves, separate to opposite ends of the cell. And you can see the animation on the left. Chromosomes start in the middle during metaphase, and then the spindle fibers rip them apart. They pull them to the opposite ends of the cell. And again, this is what it looks like under the microscope. And they're being pulled apart. And then the last phase of mitosis is telophase. It's pretty much the opposite of prophase. The nuclear membrane reforms. That's these pink bubbles. Nuclear membrane reforms around the DNA. Chromosomes begin to uncoil and relax. And the cytoplasm begins to separate. You can tell the yellow part is the cytoplasm, and it's starting to stretch out because eventually it's going to snap off and become two cells. And under a microscope, you can see this one cell with the two little nuclei forming. That is telophase. During anaphase, they're separating, but they're still kind of fanned out. And then during telophase, they bunch up again. But it just looks like two little dots, but within one cell. So there's your two little dots, but it's within just the one cell. And then cytokinesis is also part of the M stage of the cell cycle. So mitosis and cytokinesis are grouped together. 4 phases of mitosis, cytokinesis gets tacked on at the end. So we talked about before, the prefix cyto means cell. And the word kinesis means to split. So this is the actual splitting of the cell in two. Like I said, it just spreads out, spreads out, spreads out until it just pops out as two, like on the left. In animal cells, the membrane just pinches closed. Plant cells, there has to be an extra step because plants have cell walls. So they form what's called a cell plate first, and that cell plate eventually becomes the new cell wall. Animal cells are on the animation on the left, plant cells are on the right with that cell plate. All right, pictures. So instead of a summary, I want you to add labeled pictures at the end of your notes. You could also draw them with each phase too if you want. Either way, you can draw them based on the pictures in this presentation, or you can print them. And I put the link on Canvas down here. It says, here are pictures of all the phases if you would rather print them and glue them to your paper instead of drawing them. It's totally up to you. You can draw them or you can print them. But that is what I want instead of a summary this week. All right. So finish your notes. The recording will be on Canvas later today. Make sure all the pictures are drawn or glued and labeled which phase is which. There is a Canvas announcement for finals. I'll go over that right now, but if you're watching the video later and you're wondering, that's where it is. And if you got here late, make sure you check your email. Check your email now for your last list of missing assignments if you want to bring up your grade. So back to Canvas. Very, very top, you have finals information. There's information in the announcement. So your final will be on all chapters we've studied so far. Chapters 1 through 5. That should say 5. Let's fix that now. Chapters 1 through 5. And then you have a list of Quizlet study sets if you want to study. And there's also more information on this document. If you click on it, it will take you to this document. Your final is 200 points. It is 10% of your grade. So if you don't take it, your grade will go down 10%. Can you afford that? So it's on all the chapters we've covered this semester. It is asynchronous, meaning you can take it when you have the time, but you have to take it all at once. Since it is on 5 chapters, you have 2 hours, just like if you were in one of the classrooms. We have 2-hour blocks to take your finals. But you can only see the question one time. That's it. You don't get to go back and correct. So you have to take it all at one time. There is no pause button. If you close it out, it will score whatever questions you finish, and I will not reopen it. So make sure you have a 2-hour chunk to sit down and take your final anytime before Thursday next week. We will not be meeting in person. Everybody's schedules are too mixed up to meet in person, but I will be available if you need help. So just email me and I can pop open the window and we can chat. You should be using your notes. You should not be using Google or ChatGPT. But study all old tests and quizzes. Write down anything you want to use as notes for your final. And then there's your list of chapters with resources linked. There's the YouTube playlist of all the lectures we've done this semester. So whether you got here in August at the very beginning of the semester or you just got here in November, you are taking the final that on all of the information we've done all semester long. So there's all the old stuff. All right. Any questions? Give me thumbs up or thumbs down. All right. So this is going to be the last time we meet. Until next semester, for those of you coming back. So make sure you take your final. If I don't see you guys next week, have a good Christmas. Have a good holiday. That's it.